Thanks everyone for coming to our open house today and for your interest in our research. I will be talking about breast cancer and more specifically how we have progressed in recent years towards a better treatment for metastatic breast cancer. And I'll start off with a short introduction into breast cancer in general. You may have heard that in her lifetime, 1 in 8 women actually comes down with breast cancer. So quite a lot of women, right? Maybe each of you has a relative or a friend to whom this happened. Out of this 1 in 8 that will get breast cancer in their lifetime, 3 of those will get what we call metastatic breast cancer. Now, what exactly is that? Metastatic breast cancer means that the cancer has moved from its original site, the breast, to other parts of the body. And specifically in breast cancer, that can be the brain, the liver, the bones, or the lung. And you can already imagine that if you have cancer in any of these very important organs, that's probably not very good for the patient. In general, breast cancer is staged into 5 different stages. The very first, stage 0, is also called ductal carcinoma in situ, or DCIS, which some of you may have heard about in the past. And that means you don't actually have a tumor yet, but some cells in the breast decided they are now going crazy and they're just growing where they shouldn't grow and way too fast. And this can eventually develop in other stages of breast cancer. Now this can actually also be detected by a mammogram. So this is very good. So early detection, really important. Stage 1, breast cancer means there's a tumor, but it's comparatively small and it's just in the breast. Stage 2 means now it has moved to the lymph nodes, the axillary lymph nodes usually first, so it's slowly starting to spread through the body. In stage 3, tumor is bigger, definitely in the lymph nodes, and it may be in other areas of the breast. And finally, in stage 4, we have metastasis, meaning now there's cancer cells in the brain, in the lungs, or in other vital organs. So let's have a look at the survival rate. Survival rate means after diagnosis, after 5 years, how many patients are still alive. And it looks actually really good for stage 0. 100% of patients are still alive after 5 years. Stage 1, 100% of patients are still alive. So you can clearly see that we have made a lot of progress in the recent decades when it comes to breast cancer. And especially for the early stages, diagnosis is so important. So everyone should always get their mammograms and their early checkups at the medical doctors, right? As soon as the cancer starts to spread, you actually see that the numbers drop. So for stage 2, we have 93%. For stage 3, we have 72%. So numbers are going down. But now if you look to metastatic breast cancer, it actually drops to 20%. So only 20% of patients that have been diagnosed with metastatic breast cancer are still alive after 5 years. And that's terrible, of course. And this is where we start our research. So we specifically focus on this group of breast cancer patients that have metastasis and try to change these numbers and get them up eventually, hopefully, to 100% as well. Now, of course, you could argue that seems rather obvious that we should help these patients. What are we doing differently in our research? And I'll tell you about the research we do here in Cold Spring Harbor and why we think we have a very new, very promising approach to helping these patients. So, if you think of an organism like the human body, you may have heard that actually it is built from small, tiny units called the cells. And within the cells, you have something called chromosomes. And these chromosomes, again, are made of DNA. Now, DNA, this is our genes, this is our inherited material, whatever is passed down from the parents to the children, right? So this is what I'm talking about when I talk about genes or DNA. Every cell in the body has that. And for the last about 50 to 60 years, scientists all around the world thought that there is a very simple 2-step process of how a gene works in the body. So you have DNA, a gene, and in the first step, it is converted into a molecule that we call mRNA. And in the second step, it is a protein. Now, proteins are generally the functional parts in the cell. They make the body run. They do all the functions in the body, or so we thought, until recently. So this is what all the scientists for the last 60 years focus on. And then, in the last, say, 5 to 10 years, we had some really cool new technologies that allowed us to find additional genes. And now these new genes they actually stop here at the first step. So they don't make a protein ever. They do not code for a protein, hence we call them non-coding RNAs or ncRNAs. And they actually are functional on this level. They don't have to make a protein. They're still very important for the function of the cell. And of course, since we didn't know that these existed, no one has ever looked into these molecules and investigated them in the context of any cancer for that matter, right? So this is all very new science. Now you may argue, well, that sounds like it's rather an exception from the rule. Is this really important for the human body? So let me show you all the genes in the human body here, and in red, only the ones that make protein, and in blue, all the other new ones that we found. So 2/3 of all genes in the human body are these new called non-coding RNA genes that we didn't know that they existed, right? So we hope that somewhere in these 2/3 of genes that we haven't explored yet, we'll find the answer, not only to metastatic breast cancer, but also to many other diseases. This part of the genome is also sometimes referred to as the dark matter of the genome, because it's everywhere, it's the majority, and we're not exactly sure what it does, right? So you can see the analogy there, hopefully. In our lab, we specifically focus on something called long non-coding RNAs. And that just means they are long, so it's defined by length. Totally easy. We also call them link RNAs. And we have about 27%, so 1/4 of the genome are these long non-coding RNA genes, right? And this is where we in our research comes in. These are the molecules we focus in and try to find a better new treatment for metastatic breast cancer. So how do we do this? We start counting them, and we found 16,000. So a quite high number, of course. And then we went through those 16,000 molecules one by one, with a computer, obviously, and try to figure out which of them are important in metastatic breast cancer. So the way we look at it is, you have a very low level of these molecules in a healthy breast, and in cancer you have a very, very high level. So the idea is, let's bring this high level down to the level of a normal cell again, and hope that this will get rid of the tumor. And that's exactly what we do. So how do we get those high levels down? We actually destroy these dark matter genes. And I'll show you how we destroy them in a short animation here. Here in the middle is one example for one of these non-coding or dark matter genes. And up here, we developed a new drug that we call antisense oligonucleotides. Doesn't really matter what they are called at this point. If anyone is interested of how they work exactly, feel free to ask me about it later. But at this point, we have this new drug that binds to our dark matter gene. There it goes. And now it triggers a mechanism in the cell that cuts this dark matter gene. And that leads to a total destruction of this dark matter gene, right? This non-coding RNA gene is now gone from the cell. So that's great, we can get rid of them. But what really happens to the cancer if we get rid of them? And the first thing we do whenever we test in a drug is we go into a test dish and test this in the lab. And I'm sure that there are some of my colleagues somewhere around that can show you exactly what we do. We use this model system called organoids, so little mini tumors that we grow. And they're out there, I hear. So you can, they're over the quick building, so if you're interested, go over, have a look at them. And in breast cancer, this is how one of these organoids looks like. And now, I hope you can see it makes these arms, right, that stretch in all directions. And this is the bad thing about cancer. They actually invade surrounding material. They invade into the bloodstream, into the brain, into the liver. So that's what we want to inhibit. Now, after drug treatment, this is how they look, right? So they completely lose their arms. Now, since they lost these arms, these protrusions, they cannot invade anymore to the brain or to the liver. So that was pretty exciting, but this is in the dish. So what happens if we put it into a living organism? And since we can't exactly test this on humans right away, the next closest model organism that we use are mouse models of breast cancer. So what happens if we put it into a mouse? This graph illustrates what will happen. So in black, the black line up here would be mice that have not received the drug. And in red, we see mice that have received the drug. And I hope you can appreciate that even from the very first dose, from the very first time point on, there's a large difference. And we actually have only half the amount of tumors in the mice that have received the drug versus the mice that didn't receive. Now you would think, well, half is not exactly 0, and you're right. But remember, this is early stage research, and we have only targeted 1 out of these 16,000 molecules at this point. So you can imagine what happens if you combine it with a second or a third. Now, remember in the beginning, I told you that the biggest problem with breast cancer is metastasis. So what happens if we give this drug to our mice? What happens to metastasis? And we're very excited to say that again, black would be the untreated mice, and in red you have the treated mice. So you actually have a 70 to 80% reduction of metastasis in these mice. And this is very, very exciting for us here. And generally, if anyone in cancer research gets a 70 or 80% reduction, this is really, really good. So we are moving forwards, hopefully soon to clinical trials, and I will take my topic with me and start my own laboratory next year. So that's exciting too. With that, I just would like to thank a number of people here. I'm in the laboratory of Dr. David Spector, here on the left, and you may have seen him around. He has a poster as well. I saw where he's explaining some of the organoids as well. Obviously, seeding through these 16,000 targets requires more than one person, so this is our lab who was very helpful with that. And we also received a number of funding from both local organizations like Stand Up for Suzanne, other than Hazard Women, and also I'm currently a Susan G. Komen Fellow, and most of our funding actually comes from the NIH, so we should thank them as well. And I would like to thank you for your attention, and I'm happy to take any questions. I'm so glad you asked the question. So in case you haven't heard it, she was asking if there's any side effects to the drug. And that's one of the really cool things about our approach. They are developed in a way that they only target cancer cells, none of the healthy cells in the body. So the hope would be that we can reduce the side effects that you currently get with chemotherapeutics, for example. Yes?
So the ASO treatment basically stops the cancer cells from spreading, but it doesn't get rid of the cell.
Right, so the question was, does the treatment stop the cells from spreading, or does it completely get rid of the tumor? So it depends on which of those 16,000 targets you target. For some of them, it only, so great, it stops spreading. For others, it actually kills the cancer cells, and then you can combine them in a way that you can get both rid of the tumor and of the metastasis. Okay, I don't know who was first. Okay, we start with yellow shirt.
Okay, so for the drug that would make the actual tumor stop spreading, how does it actually cut and destroy the...
So how does the antisense oligo, how does the drug find its target? So we are actually exploiting a mechanism that is already in the cells. So we're not introducing anything that the cell didn't have. So this small antisense oligonucleotides, these new drugs, they are very specific, binding to the target of interest that we designed it for. And then this is like, the antisense oligos is a DNA molecule, and our dark matter chains is an RNA molecule, and this DNA-RNA combination, this duplex, is recognized by the cell. So there's an enzyme waiting in the cell for exactly these kinds of structures and destroys it. And now we have blue shirt. Sorry.
Well, in your slide, you wrote mRNA at one point. Is there any difference from mRNA and just RNA?
Yes. So our friend here in the front asked if there's a difference between mRNA and RNA in general. So RNA just means all of the RNA molecules. A subgroup is mRNAs, and every mRNA molecule makes a protein. And then the other part are these non-coding RNAs that don't make a protein. But they're all from the same chemistry, right? They all belong to the big group of RNA. Yes.
Is this only for metastatic, or can you use it earlier on as soon as you discover something?
Great question. The question was if you can use it before it started to metastasize. And we're currently exploring that in the lab, if there is a benefit of giving it earlier before metastasis started, because that of course would be fantastic if you could prevent metastasis in a patient that has, for example, only stage 1 tumors, and then will be treated with this drug to prevent any spreading. So we're currently investigating, but the hope would be that eventually we'll find something that works for that too. Okay, I'm trying to remember the order. I think you were first.
We've had the human genome now decoded for
Yep.
some time. I'm wondering why we haven't seen those non-coding DNA, RNAs.
Right.
Yeah.
So why have we not found those
Yeah.
no-coding RNAs in the human genome? We have actually, but the Human Genome Project was completed not super long ago, right? And then it took some time to understand which parts of the genome are actually transcribed or processed into these RNA molecules, because it's not the whole genome, right? There is regions in the genome that are just regulatory, they don't make any RNAs whatsoever. So we needed even more technologies, even more experiments to figure out which ones make RNA molecules in the first place.
Okay.
Okay, I think there was a question in the back.
I have a question.
There's too many questions. Okay, I think you were first.
Okay, my question is, how long do you have to give the drug to get the effect?
So that's something, how long do you have to give the drug to see an effect? That's something we currently still investigate because we haven't started clinical trials, so I can't really answer it for the human case. But there have been studies in mice where you can give them, I think, up to 9 months of stability in the body. Okay, now we start with blue first. How do you get breast cancer in the first place? That's a fantastic question, really. And there is a million answers to that. So there's some people have a genetic predisposition to breast cancer, meaning it runs in their family, if that makes sense, right? So if, say, the mother and the grandmother and the aunt and the great aunt and the cousin all have breast cancer, there is the chance that somewhere in your genome there is a mutation that you have inherited as well that will eventually give you cancer. But there's also a lot of other factors. For example, nutrition or obesity is a big factor for breast cancer, right? So if you are above a certain body mass index, you have a higher risk of getting breast cancer. So I guess generally you can always say eat healthy, exercise, and I'm wrapping up. But it's a great question, yeah. Okay, we have time for one more question.
Since you've discussed breast cancer, which is mostly for women,
Yes.
is there any work going on in prostate cancer for men?
Great question.
When are we going to have a discussion on that?
Yes, no, we actually have tested some of our ASOs in prostate cancer, and the initial results look promising as well. I think I have to wrap up at this point, but I'm being around if anyone has any more questions. Thank you so much.